Okay, well, welcome to this year's installment of the educational workshop on network neuroscience and connectomics. We've got a really great lineup of speakers for this educational course. My name's Alex Fornito and I'm based at Monash University in Melbourne, Australia. And I'll kick things off by providing a general introduction to some of the basic concepts of the area. And this will provide a bit of a foundation for discussion of some of the more advanced topics that will be covered by the other speakers in this session. So to begin with, I'll just start by motivating why we might want to take a network-based approach to understanding the brain. And that really boils down to this idea that there's ultimately two fundamental principles of brain function. So one of those principles is the idea of functional segregation. And that's the idea that specific parts of the brain are specialized for performing certain tasks. This is an idea that was really most strongly developed by Franz Joseph Gall in the 1700s with his phrenological view of the brain. He ended up constructing a map like this based on certain assumptions that we now view to be pseudoscientific. But that basic idea that specific parts of the brain are involved in particular processes has been a powerful organizing force for a lot of neuroscientific research since then. And so if you jump ahead to the 1950s, you have a map like this that's in circulation. You can see that even though the sophistication of the terms has increased, the basic idea is the same. There are certain parts of the brain involved in particular functions. And more recently, with modern neuroimaging techniques, we can sort of generate these maps of specialization and segregation with increasing precision. And this is an example of an fMRI study published a few years ago where they identified the regions responsible for processing the meaning of individual words in spoken speech. So we're quite good at mapping and understanding specialization and segregation of function, but that's one side of the coin. The other kind of main pillar of brain organization is functional integration. And that refers to the idea that somehow all of these specialized processes need to be integrated in some way that gives us a coherent experience of the world. And by comparison, our understanding of integrative processes has lagged behind. It was really this kind of landmark paper by Dan Fellerman and David Van Essen, published in 1991, that helped to kick things going. So they synthesized the results of a large number of track tracing studies in macaque of the visual system. They tried to build a wiring diagram. And so you can see here, we're starting down at the retina and the LGN, all the way up through to areas of prefrontal cortex. You can see they present evidence for a highly organized, interconnected and hierarchical system. Now, a couple of years later, Francis Crick of DNA fame and the renowned neuroanatomist Edward Jones published this essay in Nature where they bemoaned the fact that we had no corresponding or similar understanding of brain connectivity for the human brain. We really didn't have many techniques for probing connectivity, and they argued that this was really a sorry state of affairs because if we were going to understand the brain at all, we need to understand how its different elements are connected with each other. And it was a auspicious time for them to be writing this because a year before we had the publication of the first demonstration of the Bold effect, which is the basis for fMRI. And a few years later, we had the first studies using diffusion tractography to map the anatomical pathways of the brain. So if you fast forward to 2005, Olaf Sporns, Giulio Tononi, and Rolf Cotter published this article in PLOS Computational Biology arguing for the need to develop a map of the human connectome. So they defined the connectome as a comprehensive structural description of the network of elements and connections forming the human brain. So there's two key ingredients to this definition. One is it's a structural description. So we're really focused on anatomical connectivity here. So essentially a wiring diagram of the brain, although the term has since been adapted to refer to kind of comprehensive maps of functional connectivity and so forth. Really, the initial definition of a connectome is in reference to anatomical connectivity. And the other aspect is a focus on a network description. And if you think about it, we find evidence of non-trivial network organization at just about all scales in the brain. So we can identify networks of molecules that are driving synaptic and cellular processes. At the microscale, we can map networks of individual neurons and synapses. At the mesoscale, we can map axonal fibers that are connecting populations of neurons. And at the macro scales that we're typically used to dealing with in human neuroimaging, we can map how white matter bundles link larger scale brain areas. Now, this kind of multi-scale organization is one of the main problems in neuroscience. How do we develop a way of trying to bridge scales and develop a comprehensive understanding of how cellular effects propagate all the way up to macro scale processes? And so ideally we would have a single common unifying framework to represent these diverse types of data. And it turns out networks, and in particular the language of graphs or the mathematics of graph theory, allows us to do that. It allows us to do that because we essentially boil the system down, regardless of its scale or its nature, to its most essential elements, and that's nodes and edges. So the nodes in the network or the graph correspond to fundamental processing units. So in the brain, you could think of these as individual neurons or brain regions. And the edges represent some kind of interaction between those nodes, whether that's some kind of physical link between those nodes or some kind of measure of functional coupling. Now, you can see that we've lost a lot of superficial features and we've really distilled the system down to its essential elements. And once we do that, we gain a lot in terms of flexibility and the sort of things we can do to understand the system. And the nice thing is that not only does this framework allow us to represent different types of neuroscientific data in different ways, but we can also use the same models to understand other diverse networks found in nature, whether they're biological networks or social networks or technological networks. So really that allows us then to embed our understanding of the brain within a much broader science of complex networks and systems more generally. And this opens up a rich variety of analytic tools and theoretical techniques. So for the rest of the talk, I'm going to focus a little bit on how we go about mapping nodes and edges and the types of analyses we can do, because these are really fundamental. And later on, you're going to hear from some of the speakers more specifically about the issues associated with defining these nodes and edges in the context of neuroimaging analyses. So we'll start with nodes, and really the way we define our nodes and edges constrains everything else. So our network is only valid to the extent that our definition of nodes and edges are. And especially with human neuroimaging, this can be quite challenging. So in the case of nodes, ideally, our intuitive concept of a node, a network node in a brain is something that's spatially constrained. So if we are going to define a node as a patch of areas or a collection of neurons, they should be spatially confined or contiguous with each other, right? They correspond to a coherent region. The elements within that node should be intrinsically homogeneous, so they should be more or less serving the same function. So again, if we take a collection of neurons, those neurons should have some kind of similar response property. If we're taking a collection of voxels, the same. They should be some kind of functionally homogeneous set of voxels. And the third property is that those nodes should be extrinsically distinct. So they should be able to be distinguished from other nodes. So the function of one node should be distinct from another node. Now we don't, I guess, one of the classic examples of a way to define brain network nodes at a macro scale is Brodmann's cytoarchitectonic map. This is trying to identify different regions based on variations in cellular architecture. And certainly attempts have been made to use those sort of approaches in the neuroimaging context and place those regional definitions in stereotaxic space, although we don't have comprehensive coverage of the brain yet. And that is only one way of defining regions, and lots of other people have developed alternative ways. So we could look at variations in chemoarchitecture, we could use anatomical landmarks, we could just divide the brain at random, or use certain functional criteria, or there's really a lot of work that's gone into data-driven parcellation, so trying to cluster brain vertices or voxels based on similarity in functional connectivity patterns or some other kind of measure. We could treat individual voxels as nodes, or recently in the Human Connectome project, there was an attempt to use multiple imaging modalities to derive a consensus parcellation of the brain. Now, each of these approaches has its pros and its cons, and really there's no single ultimate parcellation, because it does certainly depend to a certain extent on the property you're interested in. And so really it's important to think about the kind of research questions you're asking and whether the parcellation that you're using is appropriate to address those questions. Having decided on a specific way of defining nodes, we then want to map our edges. So we want to map how these nodes are connected with each other. And there's three broad classes of connectivity in the brain. So there's structural connectivity, which is the physical or anatomical connections between brain regions. Now this is intrinsically directed. So each axon has a source and a target, but unfortunately with in vivo MRI, we can't resolve that directionality. Typically we map anatomical connectivity with diffusion MRI. So our measures are fundamentally limited in that sense. We only have undirected measures. We can map which regions are connected, but not the source or target of an area. The other type of connectivity is functional connectivity. So this refers to a statistical dependence between physiological signals that we acquire in different regions. This could be directed or undirected, but the critical thing is that it's defined at the level of the measured signal. So classic examples are simple Pearson correlations between, for example, bold time series, but you could use more sophisticated measures, mutual information, Granger causality, and so on. The final type of connectivity is what we call effective connectivity. And so this is the influence that one neuronal system exerts on another. And this is usually model based. And the critical distinction here is that we're trying to specify causal interactions at the neuronal level. And so the degree to which our model actually captures these also depends in part on how direct our measure of brain activity is. So typically, if we have an indirect measure of brain activity, such as bold fMRI, we need some kind of mapping between the underlying neural dynamics that generated that signal and the signals that we measured. And so a classic example of a way of trying to model effective connectivity with imaging signals is DCM, or dynamic causal modeling, where you try to specify models of causal interactions at a neuronal level. You pass them through an observation model, which generates expected imaging signals based on those underlying neuronal dynamics, and then you compare those simulated imaging signals to your observed data. Now, there's lots of different techniques for mapping structural and functional connectivity. Depends on the species that you're interested in, and generally there's a trade-off between resolution and how invasive the technique is. So obviously with model species, we can use invasive techniques and get highly accurate measures of both structural and functional connectivity in space and time. But with humans, we are generally limited to non-invasive techniques. There are some techniques appropriate for postmortem, but most of the studies really focuses on techniques such as diffusion MRI and functional MRI. There is increasing work looking at functional dynamics in MEG and EEG, but today I'll focus mainly on issues related to diffusion and functional MRI, as these are the most commonly used in the literature. Now, generally when we're trying to map a connectome, we acquire our data, and our imaging data are noisy. And so we put those data through some kind of data processing pipeline, and that typically involves lots of different steps. And then ultimately at the other end, we build our connectome. We define our nodes and our edges, and we get some kind of map of whole brain connectivity. Now, the thing I want to emphasize is what happens in this middle part, in this data processing pipeline, has a really important knock-on effect for any subsequent analyses. And so this is just an example of some work that we've done, but it's just one of many. Lots of people have done some excellent work in this space. So for example, in both diffusion and functional MRI, head motion is a significant problem and can really contaminate our estimates of connectivity. So in this example here, we looked at 17 different motion denoising pipelines for functional MRI, resting state functional MRI. And here we plot the proportion of edges in a functional connectivity matrix that show some residual level of correlation with head motion estimates after the denoising. You can see there's quite a lot of variability. Some kind of minimal pipelines, at least a third and up to over 2/3 of the connections still have some residual correlation with motion. Whereas if you use alternative techniques, you can reduce those proportions down to below 10%. So really the way in which you process the data and the choices you make have quite an impact on how successfully you can denoise your data and your resulting connectivity estimates. And this does have a practical effect. So for example, if you're going to do a case control comparison, here we looked at schizophrenia people versus healthy controls. You only see group differences under some pipelines. And the nature of those differences is quite different. So here's a map of the connections that are different between groups. In blue, we see edges where patients showed increased connectivity. In red, we see edges where patients showed decreased connectivity. So the top is one pipeline, the bottom is another pipeline. You can see that there's a bit of a shift in the distribution of the edges. Up the top, a lot of the edges are concentrated in the center and the front, whereas with this pipeline at the bottom, they're concentrated in the back of the brain. So this is a bit of a broad brush approach, but just to highlight the fact that the way in which you process your data can indeed affect the results you get and the conclusions you draw from your analyses. So it needs to be done with care. The same holds for diffusion MRI. We did analysis looking at multiple different pipeline combinations. You know, we sort of broke down diffusion pipelines, looked at all the different choices you can make and kind of ran every plausible permutation. So in this case, we've got some 80 different pipelines, and we're just looking at the level of residual motion contamination. Again, you can see quite a large variability. In some cases, up to 70% of the connections still show some level of residual motion contamination. The good news here is that if you use the latest version of FSL's EDDI, a tool for correcting head motion, regardless of the other choices, you do a pretty good job at controlling motion level effects. For anyone interested, this is the paper here by Stuart Oldham. Now in fMRI, we have additional confounds. Motion's not the only problem, and certainly there's lots of different sources of noise, but you know, one of the two major ones are motion and also physiological noise. And it's recently been shown that fluctuations in respiration rate and depth can have quite a profound impact on fMRI signals. So in these plots, each row is a voxel and each column is a time point. And so each vector represents signal intensity fluctuations over time that are color coded. So white means high signal intensity, black means low signal intensity. You can see that there are these periods where we see global increases and decreases in signal intensity across nearly all voxels, and they correspond almost perfectly with changes in respiration depth. So this is measured using a respiratory belt. And even if you acquire all of these physiological measures and use model-based correction techniques to try to remove those sources of variance, they're not completely removed from the data. You can still see some bands here. Whereas in this work by Jonathan Power and colleagues, he showed that using global signal regression is more successful in removing these effects. Now, global signal regression in itself is quite a controversial choice and topic, and I don't have time to go into that in detail, but this is really just designed to show you that if you don't try to account for these effects, your signals and your correlation estimates are going to be dominated by these large low frequency fluctuations. And anyone interested, there's some further reading down the bottom there. But assuming we're happy with our definition of nodes and edges, the next step is to build our connectome map. So we parcellate the brain, we have our nodes, and then for every pair of nodes, we measure some aspect of structural or functional connectivity. And we take all those estimates and we build a connectivity matrix, sometimes called an adjacency matrix. So here, each row and column represents a different region. So I'm just going to focus a little bit on this matrix because that's essentially where we generate our graph-based representation from the brain. And these two are equivalent. So we're just going to talk about that in a little bit more detail. So here's an example of a connectivity matrix. So for example, this element 1, 2 represents the connectivity between regions 1 and 2. So it's 0.8. Here we have connectivity between region 1 and 3, it's 0.45, so a little bit lower. And along the diagonal, we have the connectivity of each region for itself. And in most classical analyses, the diagonal is ignored. And so really you focus on the upper triangle and the lower triangle. Right, so the upper triangle and the lower triangle. Now, depending on the type of connectome you have, or connectivity map you have, they will either be redundant or not. So in an undirected network, so let's say we've mapped connectivity using a correlation coefficient, our measure of connectivity is undirected. We're not distinguishing a source and target of a connection. We're just showing where the connections are. Then the upper triangle is going to be a mirror reflection of the lower triangle. So element 1, 2 is going to have the same value as element 2, 1. So in more general terms, Aij is equal to Aji. In a directed network, the upper and lower triangles are not necessarily the same, and the matrix is asymmetric. And depending on how you want to encode directionality, you could list outgoing projections down the columns or along the rows. But then that means the upper triangle will have one source of information about outgoing projections, and the lower triangle will have the other type of information about incoming projections, for example. So just to dwell on this equivalence between matrix and graphs a little bit more, we'll just take a simple example here. We've got here a graph of 6 nodes connected in some way, and here's the corresponding matrix representation. So for example, here you can see that node A is connected to E and C. So if we go to row A, we find an edge for column C and column E. And because this is an undirected network, we get the same if we go the other way. If we look at row A, we find a connection for column A, we find a connection for row C and row E. Now, optionally, we could use a weighted network. So the network I just talked about was binary in that all connections have the same weight, but we know that different connections in the brain vary in terms of their connectivity strength, whether it's defined using correlation coefficients or some aspect of anatomical connectivity. And so we might want to represent that in some way. So in the graph, we can represent that using variations in edge thickness. And in the matrix, we can just use different colors. Now here we've got a directed network. This is a binary directed network. And so for example, here you can see that A has 2 outgoing connections to E and C, and it also has 1 incoming connection from node C. And in this matrix representation, we list the outgoing connections down the columns. So if we look at column A, it's got the outgoing connection to node C and E. And if we look along the row, that's the incoming connections of node A, then we only have 1 from node C, right? And so you can see that in this case, the matrix is not symmetric. So here I have an element for a value for element AE, but not, sorry, for AE, but not, sorry, for EA, but not for AE, right? So they are not they're not symmetric mirror reflections of each other. And again, you can extend this to have weighted networks. Given these considerations and choices, what then is the best network to represent the brain? So as I mentioned, the simplest is a binary undirected network, but we know that brain connections vary in terms of their strength, so we'd want to represent that variation in some way. We also know, as I mentioned, that connections are inherently directed. Each axon has a source and a target, and so again, we might want to represent that, and we can do that with little arrowheads on the graph. We also know that not all nodes are the same. There's some heterogeneity in nodes in terms of their cellular and molecular composition, the functions they serve. There's a lot of interest in identifying large-scale gradients of brain organization, and these define kind of continuous variations in various different properties of brain organization that presumably has some kind of functional impact. So ideally, we would be able to account for that heterogeneity in some way. We also know that not all connections are the same. Some are excitatory, some are inhibitory. We might want to treat some as modulatory. And so in an ideal world, we might be able to represent that in some form as well. So really, our ideal model of the brain would be some kind of hybrid that incorporates elements of node and edge heterogeneity, directionality, and weightedness. And also this network would evolve through time because we know that brain networks are not static. They evolve dynamically over milliseconds and structurally over periods of years. And so we would ideally be able to capture these dynamic evolutions through time. Now, how are we actually doing? Well, most studies in the area have used the simplest type of graph, just a binary undirected connectome. We are increasingly starting to see more and more studies doing weighted analysis, so trying to incorporate weights. But again, as I mentioned, with non-invasive imaging, we are struggling to resolve directionality. So there are some techniques such as DCM and other models that allow you to resolve it in some capacity. But at the moment, most studies looking at directed networks are looking at data from model organisms where we have, for example, track tracing data that allows us to resolve the directionality. Despite that, relatively few studies are accounting for edge or node heterogeneity. So this is an area that really requires further development. And the nice thing is that graph theory does provide tools for capturing different elements of heterogeneity. And so our data just needs to catch up. So the theory gives us plenty of room to grow. And once our measurements get there, we will continue to have a kind of rich repertoire of tools to try to make sense of those data. But assuming we've kind of worked through all those issues and we're happy with our graph-based representation of the network, then it's time for analysis. And there's two broad types of analyses that we can do. So one is focused on connectivity. So that's just mapping variations in the strength and type of connectivity at each edge. And so we can do that in a kind of connectome-wide mass univariate framework, or using multivariate or machine learning techniques to identify sets of edges that are predictive of some kind of outcome. And the other type of analysis is topological. And so the topology of a network refers to any property of the network that doesn't change under some kind of continuous deformation of the network. So you could imagine if we took a graph and we rotated it or expanded it or shrunk it down, the basic pattern of connections between pairs of nodes wouldn't change. Right? And so that's the topology of the network. And so really when we're doing topological analyses, we're trying to understand how are these connections organized with respect to each other. And these sort of analyses can give us some insight into wiring principles or key important properties of brain organization. And so it's really these topics that are going to form the focus of most of the talks for the rest of the day. So first off, Janine Bijsterbosch will be talking in a little bit more detail about defining nodes, specifically in the neuroimaging context. And there's been a lot of work that's gone into trying to develop new algorithms for not only capturing valid parcellations, but also parcellations that are valid to individual people and accounting for individual variability. And so she'll cover some of that. Then we're going to have two talks on defining edges. So one from Shella Keilholz on measuring functional connectivity in brain networks and all the related issues there, and another on mapping structural connectivity in the connectome with diffusion MRI from Rob Smith. And then there'll be a set of talks focused on network analysis. So Andrew Zalesky will talk about aspects of network statistics and thresholding and null models. Bratislav Misic will talk about neuronal signaling and communication in brain networks. Rick Betzel will talk about modularity. And Sizemore Blevins is going to talk about multi-layer networks. So this provides a nice way of looking at dynamic changes in networks over time. And Martijn van den Heuvel will talk about how we can use networks to develop multimodal and multi-scale understandings of connectome organization. And then finally, we're going to talk about models of dynamics on networks. And so Petra Ritter will specifically talk about using the virtual brain simulation platform to try to understand different patterns of dynamical interactions on an underlying structural connectome. And this provides a really rich framework for using biophysical models of large-scale dynamics to understand diverse neuroimaging signals. So I hope that you enjoy the course. To summarize for this introductory talk, hopefully I've convinced you that graphs provide useful models of brain networks, particularly for understanding multi-scale organization. There's different methods for defining nodes and edges, and each of those has its advantages and disadvantages. But it's important to be aware of those because they do constrain how we interpret the findings we get after the fact, after we've constructed our networks. Brain networks can be directed or undirected, weighted or unweighted. There's different ways of accounting for heterogeneity in nodes and edges as well. And we can analyze brain networks at the level of either connectivity or topology. And each of these analyses can be conducted for specific edges or specific nodes or subsets of edges or nodes, or at a global level averaged across the brain. So the approach really does provide us with quite a comprehensive way of understanding brain organization at different levels. I'll just close with some references. Here are some great texts that are general introductions to network science, and here are some articles and books that are focused on network neuroscience in particular. Andrew Zalesky at Bullmore and I wrote a textbook on the topic a few years ago that's available through Amazon and Elsevier. Here's a link to some useful software if you're thinking about doing this sort of analysis. We also have a journal with Olaf Sporns as editor-in-chief called Network Neuroscience. It's an open access journal with very reasonable submission fees. So if you are active in the area, please consider sending your work there. And I thank you for your attention, and my contact details are at the bottom if you need to get in touch. Thank you.
Hi, thank you very much for tuning in to the OHBM 2021 educational course on an introduction to network neuroscience. My name is Janine Bijsterbosch, and I'm going to be talking about defining network nodes, how to represent the brain. So first of all, let's recap and set the stage about what we want to do in network neuroscience. We start off with a really high-dimensional, complex data set. I'm going to be a little bit focused towards resting state functional MRI data in this talk, but all of the concepts that I'm going to cover are equally important in other modalities as well. The first step that we take in network neuroscience analysis is to define a spatial summary, which is essentially going to take our high-dimensional data and summarize it into a lower dimensional set of brain regions, which are going to be our nodes in our graph. The next step is a temporal summary, essentially extracting a single time series for each node in our graph that represents the data for that node. And then once we have that, we have all of the ingredients we need to do our network neuroscience analysis, whether that's looking at all of the connections in the connectome as a whole, looking at changes in the edges dynamically over time, or looking at topological summary features of our graph. The focus of this talk is on the second step, the spatial summary step. And as you can tell, this is really the foundation of all of the subsequent steps in our network neuroscience analysis. So it's really, really important to get the node definition for a network neuroscience analysis right. Let's first of all go over some of the different concepts and terms that are relevant for defining the nodes for our network analysis. First of all, when I'm referring to a node, the goal is to identify a region of the brain that we can think of as a fundamental processing unit. And in the rest of my talk, I'm going to go a lot more over the different considerations for how we can define that. The term parcel is also used, which is essentially the same as a node derived from a brain parcellation. You might have also heard the term region of interest, which is also essentially a node, but often in a different context. Cortical area is a region of cortex distinct from neighboring cortex in function, cytoarchitecture, connectivity, or topographic organization. Interestingly, the term network is kind of confusing and gets used for multiple different things. In a way, a network is an intuitive way of saying a collection of things. And so within neuroimaging and neuroscience, a network might refer to the full connectome, so all nodes and all edges of the brain graph, as in network neuroscience. Or in some parts of neuroimaging, we refer to a subset of all of the nodes that are interconnected with each other, so a module as a network. And that kind of relates to the third definition, possible definition of a network within our field, which is essentially a macroscale system of the brain, so a resting state network, such as the default mode network. One of the features you'll see throughout my talk is this section on the right-hand side, where I list a few different relevant papers that you can look at if you want more information. If you download the slides, these links should be clickable, and so it should be easy to find these papers quickly. So here's the outline of the rest of my talk. In section 1, I'm going to go over multiple different considerations to think about when you're defining your nodes. In section 2, I'm going to go more in depth about different methods that have been developed in recent years to define the boundaries of nodes for individual people. And then in section 3, I'll discuss some challenges and caveats in terms of the interpretation in relation to how we define our nodes. So let's start off with section 1. This is an overview of the whole section, and in the next few slides, I'm going to delve into each of these different considerations for node definition in a little bit more detail. The first consideration when thinking about defining nodes is whether you want a hard or a soft parcellation. A hard parcellation, also known as a binary parcellation, essentially assigns either a 0 or a 1 for any single voxel or vertex for any single node. And so that means that a specific voxel or vertex is part of one node and not part of any other nodes. And the advantages of this is that it's intuitive and it's very easy to visualize. But the disadvantages are that it might be a bit of an oversimplification of the functional organization of the brain. And also it might not be a good fit for the noisy low SNR measurements that we get with, particularly with functional MRI, but with other MRI modalities as well. The alternative is to use a soft parcellation, which is also known as a weighted parcellation. And here, instead of assigning a voxel or a vertex as just a 0 or a 1, it can be given a continuous number or a weight. And so that means that a particular voxel or vertex might be able to contribute a little proportion to multiple different nodes. And so the advantages of this approach is that it can capture more complex brain organization and it allows for measurement error. So essentially by allowing a little bit of fuzziness in the borders, for example, you can perhaps better capture noisy measurements. The disadvantages of this approach are that it can be a little bit more difficult to interpret and visualize. And in some cases also it can be more difficult to compare across different studies. And that's because the methods used to define soft parcellations are typically performed on the data set itself. A common example is, for example, independent component analysis. And so the breakdown into different weighted networks might be subtly different across different studies, making them a little bit harder to compare. The second parcellation consideration that we're going to talk about is whether nodes should be contiguous or not contiguous. So a contiguous node is essentially a single blob of spatially interconnected voxels or vertices, whereas a non-contiguous node contains multiple different blobs in separate parts of the brain that are not interconnected. The advantage of a contiguous nodes is that it fits most closely with our conceptualization of the brain as a graph, and you can draw lines between different areas in the brain. However, the disadvantages are that it can be inappropriate for some edge-based measures, such as partial correlation. An example of this is if you take the bilateral primary motor cortices, for example. Whenever you calculate using partial correlation, the connectivity between the left primary motor cortex and, for example, the supplementary motor area, you would regress out the time series from the right primary motor cortex. And if these regions are super similar to each other and have a very high correlation, which is commonly the case, then you end up removing essentially all of that shared signal, and you might be looking for connectivity in the noise space rather than focusing on interesting signal. On the other hand, advantages of non-contiguous parcellations are that it can capture more complex organizational principles in the brain, for example, bilateral organization or network organization. However, one of the disadvantages are that you lose the ability to study within node connectivity. So for example, if you combine the left and the right motor cortex into a single node, then you can no longer study connectivity between those two regions. The next consideration that we're going to talk about is dimensionality, or how many parcels do we want to cover the brain? And so the question that you might want to ask there is, well, tell me what is the best number of parcels? And the answer to that is that we don't know. We don't have access to the ground truth functional organization that would give us the right answer to validate methods against. And it's useful to remember here that the number of parcels is to some degree limited by biological and physiological limits in terms of the brain and in terms of our measurements. So for example, when we're using resting state functional MRI, what we're measuring is a hemodynamically mediated signal. And so the vasculature has an influence on the parcellation that makes sense at that level of measurement. It's also useful to remember that all data-driven solutions are optimized. So essentially whether you use independent component analysis or a clustering approach, all data-driven methods are trying to optimize and find the best possible solution given the constraints of the model. So in a way, all parcellations are appropriate and correct summaries of the brain. The last thing to remember is that there's probably no such thing as a single best dimensionality for the brain, because the brain is organized hierarchical in that you have, systematic organization within individual layers of cortex, but also entire parts of cortex and cortical areas that have the same sensitivities. So there's probably multiple different levels of the hierarchy that accurately represent the brain's inherent organization. So how do we gain insights into this? Well, if you're interested in studying these sorts of questions, I think there's a lot of opportunities for using hierarchical models to link different scales of analysis to each other more explicitly. Multimodal research, leveraging different modalities to gain further insights beyond the limits of the measurement of MRI. And potentially testing stability and not homogeneity, because homogeneity within a node is always going to improve as the node gets smaller and smaller, but that's not necessarily what we're aiming for. And so perhaps a better question to ask is, what is the best dimensionality for your specific study that you're about to do? And so for that, I would recommend to try and think about your research question in relation to the decision of the dimensionality. For example, if you're interested in connectivity of the posterior cingulate, you probably don't want that region to be part of the default mode network as a whole as a node, but you probably want it to be its own node so that you can measure its connectivity in itself. Another thing that you can do is test whether your results are stable across multiple different dimensionalities, so that it's not just a fluke result that you only see with one dimensionality, but it's the inferences that you're drawing are somewhat stable across at least a small range of different dimensionalities. And I would advise you not to just use the same parcellation that your lab always uses without thinking about it, because it is the case that for different research questions and different end goals, you might want to use different dimensionalities and different node definitions more broadly. The next consideration that we're going to talk about are the characteristics of the sample of your study. So there are a lot of atlases or parcellations available out there in the literature that you could just pick up and use. And actually, towards the end of my talk, there's a slide that lists a few of them that you can look at. So there is an advantage of using these high quality available atlases in terms of having really good generalizability to other studies that have been performed, and really good replicability if you do a similar study in, for example, a different cohort or different patients. However, the disadvantages to be aware of are that these atlases that are available in the literature are typically based on young, healthy subjects. And so if you're studying children or older people or patients with a particular disorder, then you might want to think about whether the parcellation that you're using is really appropriate and likely to be a good match to the population that you're interested in. And so an alternative option is to use the data that you have that you're going to use for your study to do a study-specific parcellation. So for example, you can use some of the algorithms that have been shared using, for example, either independent component analysis or different clustering approaches that people have developed to actually derive a parcellation that is specific to your study. And so the advantage of that is that you're going to get the best representation of your data. But a potential disadvantage of that is that it can be a little bit harder to relate your results to other papers because the nodes might have shifted a little bit. The last consideration that we're going to talk about is the imaging modality that you're working with. Network neuroscience analysis gets done using resting state fMRI, diffusion data, and other modalities as well. However, the nodes from one modality are not necessarily the best option to use in a different modality. And that's because the borders of structural organization don't always align particularly well with the borders of functional organization in the brain. And so what I would recommend is to use a parcellation that's informed, whether you do it on your own data or whether it's from the literature. Use a parcellation and a node definition that is informed by the modality for your study. It's also a good idea in general to include specific justifications for your choices in this category and in all of the other considerations that we mentioned as well. And one specific piece of advice here is not to use atlases such as the AAL and the Harvard-Oxford Atlas for resting state network neuroscience analysis. And that's because these atlases have been derived on usually quite a small number of subjects based on anatomical landmarks. And it's been shown that those boundaries in those regions don't match particularly well with functional organization and resting state data. For example, the first paper in the list of relevant papers shows that a little bit in more detail. So in summary, for section 1, I would really encourage you to take the time to think about different node definition considerations, all of the different options that we've listed here and all of their advantages and disadvantages, and make an informed decision for your study. If you rush in and just want to get to the interesting stuff for network neuroscience, you might end up shooting yourself in the foot, because how good the interpretations are really stand or fall with how well designed your nodes are in the first place. It's also really helpful to include a justification of your choices in the paper. As I've shown you, there's almost never a clear right or wrong answer. And so saying what you did and why you did it can really help people understand. And the last thing that you could consider is testing multiple different node definitions. I've mentioned this already in relation to dimensionality, but you could also consider a weighted and a soft parcellation and see how that might change your results and their interpretation. I've talked a lot about how important nodes are for meaningful results from network neuroscience analysis. And one of the things that myself and many others in the field have realized in recent years is that there's a lot of systematic variability in the functional organization from one person to the next. And so that means that if we define our nodes and our parcellation using group averaged or group concatenated information, then those boundaries of that group level parcellation or that group level node might not actually match up very well with the individual level organization. And that's really important, because when we then extract a time series, an average time series within a node, for example, we might be mixing together all sorts of information from different brain regions. And so I want to focus this next section on different methods that have been developed to improve our ability to define nodes that are meaningful, with meaningful boundaries at the individual subject level. This is still a fast-moving field, and there's a lot of new research coming out in this field, but I think it's really important to be aware of how people are thinking about this challenge and what sort of directions people are taking to try and address this problem. The first approach that we can think of for improving our ability to have meaningful boundaries for individual subjects is essentially to improve our registration. So if we do a better job at aligning the brains in the first place, then those group defined boundaries might be more meaningful for each of our people, for each of our subjects. And so on this slide, I'm summarizing two very different approaches to improve registration. The first one is called MSMO, or multimodal surface matching, using all different modalities. And so here the idea is that you can, compared with volumetric registration, you can already get much better registration on the surface. But typically in FreeSurfer, for example, that's only driven by structural information. But actually, if you combine that with resting state information, task activation maps, then you can do a much better job aligning across people those functional differences. And so an advantage of this approach is that it can get really measurable improvements in the sharpness of group average results. So that's shown in the figure on the left here, and it's also shown in the Coulson paper that's listed in on the right part. However, an important challenge with this approach is the known existence of non-diffeomorphic variance across people. So for example, if you have a brain region that's here in one person, but then in another person, it's kind of split up into two regions, and there's another region that kind of goes in the middle of them, then that sort of variation is not something that can ever be captured using smooth warps. The second challenge is that potentially we're losing meaningful information by removing the spatial variability that exists in the first place. So for example, we've shown that those warps that get estimated for MSMO, which are usually just used to transform brain data into a common space and then discarded, those discarded warps actually are really significantly associated with behavior. So it seems that there is some interesting information there that we might be removing. A second and completely different take on the problem of registration is the idea of hyperalignment. And here the method essentially does away with brain space completely, and it doesn't try to align patients in terms of their voxels or vertices, but instead it projects people onto a shared space that isn't the spatial brain space. So you can align subjects by projecting them based on shared activity or brain activation or shared connectivity features. And so the advantage of this is that it gets around these non-diffeomorphic variants because it's no longer trying to align the brains as a whole. However, challenges with this is what information do you use to drive this projection? How do you constrain it? And how do you interpret this? Because we're used to kind of thinking about graphs and kind of making figures that project graphs back onto brains. So another approach that people have taken to be able to investigate more individualized nodes is what I'm calling deep phenotyping. And here the idea is essentially to do away with the group level completely and analyze data at the individual level. And this is done by obtaining hours of data for each individual so that you have sufficient degrees of freedom and sufficient data to be able to robustly estimate a data-driven parcellation just for that individual. And so the advantages of this approach are that it's by far the best way to capture truly individualized nodes. And it really fits with the goal of personalized psychiatry, being able to scan one person and draw interpretable conclusions from just that data set. However, some of the challenges with this is that it kind of lacks correspondence. So often what we end up wanting to do is compare different scans to, for example, compare patients versus control or look at associations with behavior or with symptoms. And for that, you got to be able to compare apples to apples and oranges to oranges. And when you're running a completely separate parcellation for separate individuals, that correspondence of being able to say, is this node in person 1 the same as this node in person 2 can be challenging when you're running these analyses separately. And similarly, generalizing your results to other papers can be challenging, but most importantly, one of the challenges with this approach is high data requirements. It might just not be feasible to get hours of data for because your patient can't tolerate it, or because you can't afford it, or because you don't have the kind of infrastructure to be able to do that. The next category of methods that have been developed to identify individualized node boundaries is to essentially do a post hoc mapping of a group-defined parcellation to kind of redefine each of the nodes based on each individual's data. And there's a number of different methods that fall within this category. Dual regression and back projection are options for independent components analysis defined nodes. But there's also options for machine learning and iterative regression. And so the advantage of this approach is that you make sure by doing the group parcellation in the first place, you make sure that you have correspondence across subjects. And so that makes it easier to compare between different subjects. And another advantage can be that some of these methods can be more easily compared across the literature if you're using the same starting point. However, some of the challenges are still these non-diffeomorphic or non-overlapping variants. If a node is in a specific location at the group level, and it's in a completely separate location in an individual subject, then most of these methods will struggle to find it. And that kind of links to the next challenge with this approach, which might be that some regions might end up being missing in some methods, or actually being made up. So if the post hoc projection is too strongly driven by group level information, then it might find a region regardless of whether it's there or not in that individual, or if it's too strongly driven by the individual data, you might end up with missing regions, which then becomes really hard to deal with later on within the network neuroscience analysis. The last group of methods that try to get at individualized nodes are integrated models. And so these models are designed to, within the same hierarchical framework, estimate both a group level solution and an individual level solution. And this is typically done within a Bayesian framework, iteratively optimizing both of these aspects and using some group priors to constrain the individual estimates. advantages of this is that it's a really nice mathematically principled hierarchical model of different types of variability. And of course, we do get that correspondence across subjects because there is some constraint of using the group level information to drive the individual level estimates. However, that's exactly where the challenge is. There's a lot of different parameters in these models, and how do you determine, for example, the appropriate strength of the group prior? You want to make sure that it's strong enough to get that correspondence, but that it's not so strong that we're losing out on finding meaningful individual variation in the nodes. And so to summarize this section on individualized nodes, I would strongly argue that the interpretability and meaningfulness of results from a network neuroscience analysis are much better when we can think of nodes as being meaningful matches to the data at the individual subject level. However, this field of how to best do this is really a moving and developing scientific field. So I'd encourage you to inform yourself about what research has come out recently and what are the options for defining individualized nodes. And although there's no clear best solution, it's probably better to try and do something to get more accurate subject-specific boundaries compared with just using group level boundaries. And lastly, as I had as a takeaway in my last summary, it's really useful if you can include a justification of your chosen method and how you chose it and how you weighed up the different advantages and disadvantages. So in the last section of my talk, I want to discuss a few different gotchas or challenges in relation to interpreting network neuroscience results that come out of these, the definition of the nodes. The first interpretation challenge that I want to talk about is oversplitting. And this is related to the topic of dimensionality that I discussed earlier. And the thing to be aware of here is that our definition of nodes and the dimensionality of our nodes interacts with the method that we use to define our edges to build our graph. So for example, in this figure on the left here, you can see that if region 2 or node 2 is really one homogeneous region, but accidentally for one reason or another, we end up splitting it into region 2A and region 2B, then we end up when we look at the edges, missing this connection between node 1 and node 2 when we do partial correlation. And that's because when we estimate the connection between node 1 and 2A, we regress out 2B and vice versa. And so that's an example of this node definition leading to false negatives. This is challenging to get exactly right, and therefore one of my recommendations is to, again, include a clear justification of your node definition that matches and is a good fit to your research question, and also avoid overinterpretation of the results and discuss potential caveats. It can also be useful to investigate both full and partial correlation so that you can get a better sense of what information is shared between them and what information is different between those. The second interpretation challenge that I want to talk about is node heterogeneity. And here, when we do network neuroscience analysis, virtually in all possible analysis, we define a single time series for each node. So we represent each node as a single averaged or otherwise regressed time series. However, this is highly likely an oversimplification of the complexity. And so there might be inhomogeneity or heterogeneity within the node. So some recommendations for looking into this further is, first of all, to consider a soft parcellation instead of a hard parcellation, where we can portion out different amounts of variance within the same vertex to contribute to one node and to contribute to another node. Another recommendation or potential option here, if you're interested in this heterogeneity, is to consider doing a gradient analysis to explicitly investigate different kind of modes of organization within a node. And again, I'd encourage you to look at some of these papers on the right here to learn more about this. And the last challenge in terms of interpretation that I want to touch on briefly is representational ambiguity. And here the idea is that when we define our nodes, it essentially determines the lens that we take to look at our data. And to some degree, it limits the types of results that we might get. An important thing to be aware of is that we have shown in multiple different papers that connections or edges between nodes can be influenced by other sources of variability that are not necessarily interpretable in terms of connectivity or coupling. And examples of these are amplitude changes or unmodeled spatial variability. The recommendations to kind of counteract that would be to use, again, individualized nodes, define boundaries of nodes for each individual using that person's data to limit these unmodeled spatial variability influences. And another option that might be really interesting to look into is to use a technique called additive signal change analysis to test for amplitude influences. And that relates to the first paper on the right-hand section. So in summary for this section on interpretation challenges, I'd encourage you to be careful not to overinterpret results from a network neuroscience analysis. There's a lot of caveats in terms of how we decide to partition of the brain that can affect the results that we see. One interesting option is to run additional analyses to test for influences of different types of variants so that you can better understand your results and draw more informative conclusions from your results. And the last recommendation here is to discuss potential sources of ambiguity in the paper, which can be really helpful to further the field and inform a broader discussion about this. So as I said at the start, I have a table here with different resting state atlases. This might not be completely comprehensive, but this covers some of the most commonly used ones. And again, these links to these different papers are clickable if you download these slides. And so if you're looking for using a literature-based atlas or a resting state atlas parcellation, then this is a really good starting point to look at. I've also included a short self-study quiz here. I'm not going to run through these questions, but I would encourage you to look at this because considering some practical examples can be a really good way to learn. And you can find the answers as well as a short explanation on the bottom. Again, there's not always necessarily an obvious right or wrong answer, as you can see throughout my talk, but there are, when you're weighing up the advantages and disadvantages, there's usually a preference for one or the other. And so that's really what this quiz is trying to help you start thinking about. So with that, I want to say a big thank you to the organizers for this educational course, Andrew Zalesky and Alex Fornito. Shout out to my lab, my funding, and my wonderful collaborators. And thank you very much for your attention. And please do join us for the interactive sessions to come and chat to us more if you have questions, but also if you don't have questions and you just want to have more of a conversations about some of the topics that we covered today. Thanks.
Hi everybody, I'm Shella Keilholz, and this part of our educational course will go over ways to measure functional connectivity in brain networks. Here's a quick overview of the topics we're going to cover today. The first is average functional connectivity, where you calculate metrics over the course of the entire scan. And there's going to be a special emphasis on correlation, because a lot of what we'll discuss in terms of caveats and interpretation for correlation also applies to other methods. And then we're going to talk about time-varying functional connectivity, where you get metrics that vary over the course of the scan. And there are a variety of approaches to this. I've broken into a few different categories. At the end, we'll have a brief discussion about summary metrics, and then we'll talk about null models and validation. Let's start with the average functional connectivity. So when you're talking about resting state fMRI data, you could think of characterizing activity within an area or coordination across areas. Now, there are methods that look at the amplitude of these low frequency fluctuations that characterize activity within an area, but I'm not going to talk about those today. For the topic of today's talk, we're going to stick to areas that talk about coordination across different brain regions. And this approach was originally taken because when we have these intrinsic resting state fluctuations, the signals are very low. And we don't have any known timing of tasks or stimuli to guide us so that we can average and get rid of some of the noise. So instead, the idea is that if you have neural activity in two different areas, That leads to metabolic changes in 2 different areas, that leads to hemodynamic changes in 2 different areas, and the bold signal in 2 different areas. And so if your original neural activity between those 2 areas is correlated because they're communicating somehow, you should get correlated bold signals out. And this is kind of the basis of the whole idea of how functional connectivity works. Now, we all know that in addition to neural activity, there are a lot of things that can influence this kind of chain of events that leads us to the bold signal from the neural activity, and that can also introduce correlation across areas. So we have to be careful to keep those in mind when we're trying to interpret our data and make sure that there's not a way that those are confounding our results. For example, if you have head motion that's different across groups, you might get a difference in functional connectivity. Correlation measures the way that signals go up and down around their mean. If they go up and down around their mean together, so that they're both going up at the same time, correlation is going to be high. If one goes up when the other goes down, it's going to be anti-correlated. And if there's no relationship, it's going to come out to be about 0. So this is a measure of linear dependence. And depending on what you're looking at, you can calculate the correlation between time courses from different voxels, different regions of interest, or even between networks. Now, I have an illustration over here that I took off of Wikipedia because I think it gives a really nice demonstration of correlation. So this top row is kind of what we think of when we think of correlation. So if we have a signal that has a correlation of 1, you have a perfect match between when one signal goes up and the other signal goes up, we get this nice straight line. A correlation of 0.8 has a pretty clean relationship. You get to 0.4, it's getting a little iffy. I mean, you can still see the trend there. 0, there's no relationship between the 2 signals at all. It's right here. And then, of course, the negative, the anti-correlations, have the same sort of general shape, but the relationship goes in the other direction. Now, note that in the second row, all of these except this middle signal have a correlation of 1 or -1, even though the slope of the relationship between them is different. And that illustrates that the slope really doesn't matter for correlation. All that matters is whether there is a 1-to-1 correlation between the deviations from the mean in one time course and the deviations from the mean in another. And we mentioned that this is a measure of linear dependence. So if we look at this bottom row, I mean, clearly there's structure in all of these data sets, but the correlation value for all of them is 0, and that's because correlation is not good at picking up this kind of nonlinear dependence. So I think there's yet another good illustration of this at the bottom. Again, kind of in the top left, you have typically what we think of, where you have these sort of scattered points around a line that we calculate a correlation value for. Again, because this is a linear measure, when you look over here and there's clearly a nonlinear relationship, you don't pick up the right relationship. But I also want to point out that correlation can be driven heavily by outliers. So in this bottom row, these signals are, perfectly correlated except for 1, and so you get a big change in correlation there. But more importantly, I think, over here, there's no relationship at all for most of these points. You've got 1 out here that's driving the whole calculation of correlation. We saw in the equation for correlation that we're looking at the relationship between the normalized time courses. And so that means correlation is not affected by amplitude. And here you can see that I have signals with 2 different amplitudes, but they're clearly the same signal otherwise. And of course, the coefficient of correlation is 1, despite the difference in amplitude. Now, what does matter is noise. So if we add noise to those signals, now I'm taking the same signals and just adding 1/2 sigma Gaussian noise, our correlation coefficient drops to 0.58. And if I add more noise, the correlation coefficient goes down further. So while amplitude is not important, the signal to noise ratio of the signal definitely is. There are two common approaches to using correlation in practice, and the first is seed-based correlation. This is most frequently used if you already have a hypothesis about a particular area or circuit that might be involved in what you're interested in. And for that, what you do is you take a seed area, it can be a voxel, it can be a region, you take the time course from that area, and you do correlation between that and the time course from every other voxel in the brain. So I'm showing a seed-based correlation map right here with the seed up here in the frontal area. You see that's the very high correlation value, and every other voxel that's significantly correlated to that seed is overlaid on color on top of the anatomical image. Another way that you can use correlation, especially for, let's say, you want to look at the whole brain or you don't know which area might be affected by the changes you're trying to study, is to do some sort of parcellation. So you can take your brain image, you can register it to an atlas or find another parcellation scheme and basically divide it into chunks, right? So, this is a very ugly brain parcellation, but I could divide this into different pieces. And for each of these areas, I can average the signals from all of those voxels together to get a time course. And then I do correlation between the time course from that area and the time course from each of these other areas. And this is usually summarized as a matrix. So you have one row for each of your parcels and a column for every parcel that it can be correlated with. So these should generally be symmetric so that the top triangle of the matrix and the bottom triangle of the matrix are the same. Now, the exact symmetry can depend on how you set up the matrix, but basically you only need half of those data because the correlation between parcel 1 and parcel 3 is the same as the correlation between parcel 3 and parcel 1. So here are some examples of seed-based correlation maps showing different networks. This is from rat data now, but in this top row, we've used a seed up here in somatosensory areas, and you can see kind of the network of correlation you get. If you move the seed in the same data to the hippocampus, we get this bilateral hippocampal areas. Again, if you move the seed to the caudate putamen, you get those areas, etc., etc. So if you want to do this on the group basis, what people usually do is they register all of their data to some sort of atlas first. Now, I'm not going to go into detail, but you can imagine that if you line all of this data up in common space, you can concatenate the data, and then you can do correlation between a seed that you select and all of those other time courses at the group level. And there's a lot of ways to do this, probably too many to go into right now. So going back to correlation matrices then, if you want to look at a difference across conditions or across groups, the most common approach is to make a correlation matrix for each group and to do some kind of statistical test between the two matrices to identify all of the areas that are significantly different. And so, that's shown over here on the right where most of the entries are green, meaning they're not significant, and we have a few that are significantly negative or positive. Now, normally this matrix would be symmetric. I'm actually using one that we did for something else that has a different treatment for the bottom half, so you can just ignore that part. So if we do want to do statistics, one thing that you have to remember is that correlation is not normally distributed. The maximum correlation you can have is 1. The minimum correlation you can have is -1. And so if you want to do statistical analysis that assumes a normal distribution, you've got to kind of modify those correlation values a little bit before you do the statistics. And the way that most of the imaging community uses is called the Fisher transform, where it's just the hyperbolic arctangent of the correlation coefficient. And so if you look over here at the right, you can see what that does. It takes these correlation values that are close to 1 and it kind of stretches them out towards higher values, and the ones that are close to -1 get stretched out to more negative values to make the distribution more like a Gaussian. So when we have one of those correlation matrices, let's just say we have a parcellation into 360 areas, like you might for the Glasser parcellation, then your correlation matrix is 360 by 360, which means you have over 100,000 correlation values. And so if you're looking for differences, you're looking for differences at over 100,000 locations. Now, in this little calculation, I've ignored the fact that the matrix is symmetric and you'd really only look at half of it, but still these are huge numbers. Anyway, if you have that many values and your threshold for significance is set to 0.05, just by chance you would expect to have over 6,000 significant values. So then how do you decide if the values you see are real or if they're just popping up because you're doing so very many comparisons that you would expect to get these just by randomly? So there are a lot of different strategies. Again, I'm not going to go into all of these. I just want you to know that they exist so that you have a chance to go out and look into them. One is called Bonferroni. That's really the strictest. Usually you take your significance level and then divide by the number of comparisons to set a new level that you used as a very strict threshold for significance. Another possibility is clustering, because you can imagine that if you have those 6,000 significance values that occur by chance are probably kind of randomly distributed, right? You wouldn't think that they should have a particular spatial structure. And so if you have 5 of those values that are located right together, the chances of that are lower. And so you can use a combination of the statistical significance threshold and the likelihood that they occur in proximity to each other to calculate some sort of significance based on the cluster size. And then there are many other approaches that people have developed that you can also use, and I've listed just a couple here. Since we've gone into detail for correlation, I just want to summarize a little bit before we move on to other methods. So we know correlation captures linear relationships, may not pick up nonlinear structure, and can be driven by outliers. The nice thing about correlation is I think compared to some of these other methods, it's relatively easy to understand where differences between groups might come from. You can think about, what affects correlation values and how that might occur as a difference between the groups. We pointed out some factors like changes in the signal to noise level, which could come from vascular changes, from levels of motion, other effects that are different between your groups, and these can alter correlation values. So you need to kind of be cautious. Correlation serves as the basis for many subsequent analysis methods, especially people who do a lot of work with graph theory, which we're going to talk about briefly later. And so anything that you do with those subsequent analysis methods are subject to all of the caveats that we have with the correlation measurements themselves. So I like to say we should interpret this with caution. At a minimum, we want to make sure that, things are as much the same across groups as they can be. The amplitude and the frequencies of the bold signal, the levels of motion, and possibly global signal. I know that's still controversial and it's hard to tell where it comes from in some cases, but that also is something that could possibly impact the overall levels of correlation. The other technique that's widely used for average functional connectivity analysis is called independent component analysis, or ICA. And this is performed by several different algorithms. And the idea is to separate some sort of multivariate signal that is your resting state fMRI signal into sources of independent non-Gaussian signals. And this can be based on mutual information, or they can try to maximize the non-Gaussianness. And there are different names for the different algorithms that you'll come across. For a resting state fMRI, this is usually applied spatially so that areas that have similar signals are grouped together. And so you see very similar results to what you get with correlation. You can get the same networks with both techniques. Now, when you have to run an ICA analysis, you need to decide how many of these components you want. And that's not something that ICA can really tell you. It can't say, well, how many components should there be? And unlike PCA, it doesn't automatically tell you how strong the components are. So for example, this is an ICA analysis of some resting state data. And each of these components in this case is more or less a network. So you can see different different ones shown here. Let me find, here's somatosensory network, and we're looking down the column to look at different brain areas in that network. Now, something to notice is if you look here, this is what they're calling the PCC network. And you see a lot of the posterior cingulate and those two slices near the top. But over here, we have the default mode network, which looks pretty like the default mode network, but doesn't have a lot of those PCC components. So I think this is an example of how ICA can maybe split something into different components if you ask for more components than you really want. And, you know, at the same time, we could have taken the other extreme and gone for even more components and tried to separate basically each of these little blobs into its own component. So that's another way to do it. So there's not one way that's appropriate. You might want to set your components to try to pull out networks or to try to pull out areas. It really depends on your study. To summarize for ICA, what I think is really the greatest thing about it is that it's completely data-driven. And so you don't have to pre-select a seed region or anything like that. Now, of course, that's also true for your correlation-based matrices when you do a brain parcellation as well. And like the seed-based or the correlation matrix version, you can do ICA on concatenated data, and that way you can get group results. You can follow ICA up by another type of analysis. For example, sometimes if people use ICA to kind of parcellate the brain into those different brain areas, they might do correlation between those parcels. The cool thing about ICA that I hadn't really mentioned yet is that sometimes it can separate out noise components. And so this is part of the fixed pre-processing algorithm where you do ICA and you look for components that look like known sources of noise and throw those out. Now, it assumes spatial independence, which is not likely to be true. How big an effect that is, I don't really know. But what I do think you have to consider when you're doing an ICA study is that differences in the components that you get across groups can be difficult to interpret. So let's say you want to look at ICA components in Alzheimer's versus a healthy control group. Well, you can put all of that data together and run your ICA, which will force them to have the same components. So that makes them have the same spatial structure. Or you can run the ICA separately on the two groups, but then your components may not have the same spatial structure, and you have to decide how you're going to compare them. So there are many other measures people have used for average functional connectivity. Things like partial correlation, where you basically regress out the influence of a third area to help reduce the effect of common inputs. You can use mutual information to get dependencies that are not necessarily linear. There are frequency-based approaches where you look at coherence that allows for phase differences between areas. So, there's a lot of literature existing on these, and a great place to start is this paper by Steve Smith on network modeling methods that will give you an idea of how some of the common ones work. So now we're going to switch from average functional connectivity to time-varying functional connectivity. So in contrast to average techniques that give you one metric of connectivity, like correlation for the whole scan, for time-varying analysis methods, we're going to get multiple metrics over the course of the scan. And a simple way to do this is to take your 10-minute scan and divide it up. Maybe you segment it into 1-minute windows. Or maybe you do something like take 1 minute and a 1-minute window and slide that along your scan. That's called a sliding window technique. Or you might say, well, maybe I need to use different window sizes, and I'm going to change my window size based on something that I see in the data. That's called an adaptive window approach. And there are other methods that build on individual events, so things that happen at single time points, or repeated spatiotemporal patterns, where you have kind of a spatiotemporal sequence that recurs throughout the scan. And all of this is a very active area of research, so we're only going to scratch the surface today. Like we did for average functional connectivity, I want to kind of use correlation to talk about time-varying connectivity. And so one of the very simplest time-varying approaches you can imagine is to do sliding window correlation. So in this case, we have a time series from one area of the brain and the time series from another area of the brain. And what we do is we take those time series and we take a window, that's this gray area, and we calculate correlation for that section of those two time series within those windows. Maybe it's 0.5. So I'm just making it up. And then we slide that window to the next point and we get a new correlation coefficient. Maybe it's 0.8. And so you get this time series of correlation. There you see it slide. This is what a time series of correlation looks like. One thing to notice is it depends on how long you make your window. So this legend here shows that we have 3 different window lengths, one 25 time points, one 50, and one for 100. The longer window, the 100 time point window that's in black, is much smoother and less variable than what we see in the shorter windows. So the 25 window has a lot more of these jagged peaks and things because you capture more of this variability with those very short windows. When we started doing sliding window correlation, part of what our rationale was is that we thought maybe areas that looked like they were not correlated over the course of the whole scan might be correlated for some portion of the time. And so we did these sliding window correlations. We took those time courses we had and we divided them into times of positive, moderate, and weak connectivity to make kind of these histograms. These are shown as bar plots in this particular plot. And we looked at these areas, which mostly don't show strong average functional connectivity. We said, wow, if we look at those, sometimes we see that these are strongly correlated or strongly anti-correlated. Cool. But then we looked at areas that we obtained by matching time courses from different rats. And we got the same sort of distribution. And so that made us worry a little bit that maybe this variability that we were seeing wasn't really as meaningful as we would like. We know that the resting state fMRI signal is band limited and autocorrelated, and we thought that might set up, the possibility of finding this kind of structure even in randomly matched time courses. So to understand that better, we did some network modeling where we made networks where we knew what the underlying time varying correlation was. That's shown by this black line in these graphs. And then we measured what we got with different window lengths, and those are shown in the colored lines. These were starting with 25 TRs at the left and going up to 200 TRs at the far right. And what we found is that no window length really provides a good estimation of correlation, although you do better if your window length is comparable to the length of time that a particular state persists. So, that made us worry about, well, were we getting anything useful out of sliding window correlation? And so we did a study where we looked at the correlation between band-limited power from 2 electrodes, where we acquired the LFPs at the same time as the bold signal. And so we plotted the correlation between band-limited power over time, the correlation between the bold signal from the 2 areas over time, and then we looked at how closely related those 2 were. And we found significant relationships between the time-varying correlation in the bold signal and time-varying correlation of power in bands like theta and delta. So that encouraged us that there was actually some neural basis for this type of variability. In humans, the same sort of thing can be seen behaviorally. If you look at the sliding window correlation between the default mode network and the task-positive network, you can get something that distinguishes between people who are fast performers and slow performers on a particular reaction time task. Now the details of that don't really matter, but what we're saying here is that there is a link between metrics we see with sliding window correlation and things that are linked to neural activity or behavior, so that we must be getting something meaningful out of it. So, I think for a while there was kind of a big debate about should we use sliding window correlation. There were studies saying we shouldn't, other studies saying that, this is great, it works well for distinguishing patient populations and things like that. And I think what we've come to is that, it has its uses. I think its performance depends strongly on the signal to noise ratio of your signals. And it's possible that if we do sliding window correlation, say, of the neural activity and the bold signal, we're kind of distorting the signal in the same way so that we're preserving some information even though, the actual sliding window correlation itself may not be terribly accurate. So I'm running a little short on time, and I'm just going to briefly touch on a couple of these other dynamic analysis techniques. One is the idea of using individual events. So here you pick an area of interest, for example, you set a threshold, maybe here at an amplitude of 0.6, and you find all of the times when the signal in that particular area crosses that threshold. And then maybe you cluster frames of the image based on those time points, or you look at the average activity pattern. There are a lot of different ways to do this, and there are some great groups working on it. You can look at work by these authors. And the reason this is cool is because people have shown that a lot of what we see as correlation is really driven by individual events. So here's a figure from Liu and Dunn showing that if you have a single frame, it might look like this row at the top, and you can average those frames that look alike over a single run, get something that's a little bit cleaner, and that these start to look very similar to the correlation maps that you make if you use a seed based on the same area that you use to threshold those individual event maps. So these individual events seem to drive the correlation. You can also get repeated patterns of spatiotemporal activity, things that happen over and over in the background and have a spatial and temporal component. We call these quasi-periodic or QPPs. So here's just an example of how some areas become active, others become deactivated. There's kind of a propagation over the cortex over time and a switch between the two networks. Again, I don't have time to go into these, but if you look at some of the papers, you can find out a lot more. The problem with time-varying analysis is suddenly you have too much data. You have hundreds of time points at least per person that causes a data explosion. And so we need some ways to summarize this across groups. I'm going to touch very briefly on brain states and time-varying graphs. Brain states are loosely defined as a pattern of activity that's kind of similar at different time points in the scan. I think there are still questions about what the right spatial or temporal scales are. And sometimes people do this by doing something like sliding window correlation and then clustering those results. So here's an example of brain states based on clustering from raw data. You can see here's one state at the top, which is more task positive, one state at the bottom, which is more default mode network. And then down here, we can show at any given time point which state is dominant. And you can, of course, have more than two states, and you can calculate transition rates between states and things like dwell times. You can do this similarly on those correlation matrices where your state is now defined by a matrix instead of a spatial map necessarily. Another method for summarizing the data is to turn each of your nodes, your areas, your ROIs, into the node of a graph, and then connect them with edges that represent the weight of the functional connectivity between those. Now, if you do that, you can do static or dynamic analysis on it. There's a lot of work by great research groups here. And you can calculate all sorts of metrics about how modular the graph is, the clustering coefficient, the efficiency. You can find hubs. And so these are great tools to tell you something about the overall structure of the functional connectivity in the brain. So again, something just quickly, an important area of research is finding the right statistics for a dynamic analysis. You saw how, we can get apparent fluctuations in sliding window connectivity that may or may not be different than what you would get from randomly paired data. So that approach is called a permutation, where you kind of pair data from different scans or different subjects to get sort of a random null distribution. Now, this doesn't necessarily preserve static correlation, the average functional connectivity for a pair of regions. And so it may not be the right sort of null model for your study. So really, when you're doing the statistics, you just kind of have to think about what you're trying to prove and to make sure that your statistics and your null models are designed accordingly. I just want to end with some caveats. Remember, we're looking at hemodynamics. No matter what you do to functional connectivity, we're always looking at the bold signal. It's not a direct measurement of neural activity, and neurovascular coupling can be complicated, especially in areas that we understand less well than, say, primary somatosensory areas. We have registration and smoothing during pre-processing that makes it hard to detect small areas. They can get averaged away if you're doing a parcellation. Always think about the impact of noise. We've seen how that can affect your correlation values. So you don't want to say, oh, functional connectivity went down, when the truth is really that noise went up. It's not the same thing. And anytime you do some sort of higher level analysis, it depends on what you've used for your lower level metrics. So if you're doing graph theory, it's still based on maybe correlation that you're using to measure functional connectivity between areas. And so all of the weaknesses of correlation carry over into that graph theory study. So try to be a little bit of a skeptic when you're interpreting this work. Thank you all for listening, and I look forward to discussing this with you during the Q&A session.
Hi all, my name's Rob. In this session I'm going to be presenting the fundamentals behind structural connectivity estimation for connectome construction. Now the structure of this talk is slightly unusual. I've designed it around trying to encourage a strong fundamental understanding of the technologies rather than just presenting a bunch of details, but I hope it'll all make sense as we go through. So we'll start with the basics of using diffusion tractography in this context. So fundamentally, if we have some biological white matter structure of interest, we're going to perform diffusion MRI acquisition and requisite pre-processing, and we have our raw diffusion weighted imaging data. And unlike most other MR modalities, we have this additional orientation information where the amplitude of the diffusion weighted signal is dependent on the direction in which we apply diffusion sensitization. And in this case, I'm using the corpus callosum as the exemplar, and you can see that there's a high signal when the sensitization is applied orthogonally to the fiber direction, which is from left to right, and the amplitude is small when the diffusion sensitization is applied along the direction of the fiber, so from left to right. Now, if we apply what we call a diffusion model to impose, to try to recover useful biophysical information from these data, so we have a diffusion model fit in each voxel. In this exemplar image, I'm showing the estimated white matter fiber density. And in our exemplar voxel here, I'm showing what's called an orientation distribution function, which shows the estimated density of fibers as a function of orientation. So you can see it's informing us that there's a large density of fibers running left to right and not in other directions. So this is the information that we then feed into what we call tractography, which is where we attempt to reconstruct the trajectories of the underlying white matter fibers. Now, fundamentally, what we're looking at here is we say, okay, we've got a biological white matter bundle of interest. We have our streamlines tractography reconstruction. We want to estimate some measure of connectivity that from our reconstruction data that is hopefully somehow reflective of some quantity of interest of the actual biological white matter bundle. So firstly, I want to quickly introduce streamlines tractography, and I emphasize specifically streamlines tractography here for a reason. This was the first type of tractography that was introduced, and it's used basically ubiquitously in the field, so it's the one I'll focus on today. And basically it involves iterating in a stepwise fashion through space where we sample at each location the underlying fiber orientation estimate, take a small step in that direction and simply repeat. An important little detail to note here is that while the generation of individual streamlines happens in a directed fashion, the data themselves are not considered to be directed. The streamline is equal in either direction. So in the context of graph theory analyses, these types of data are basically always interpreted in an undirected fashion. And the reason I emphasize streamlines tractography is because there is a wide range of types of tractography that are not streamlines based. Again, I won't go through the details here. It's more so that if you come across alternative techniques in the literature, you'll hopefully be better posed to identify them as such. So I now want to talk about the way that I conceptualize connectome construction with streamlines tractography, and I break it into these three steps. The first is that we need some technique by which we reconstruct a specific white matter pathway, and we derive from that some measure of connectivity. We then simply have to repeat this process for every pair of grey matter parcels within our parcellation, and we then perform some downstream analysis of those data. So what we're doing here is we're taking a brain parcellation. So on the left, I've got an exceptionally coarse parcellation. I've just got 4 lobes per hemisphere. On the right, we have a whole brain streamlines tractogram reconstruction. And we're going to combine these two things together to form our connectome network matrix. And what the contents of these matrices fundamentally are is every element in the matrix corresponds to a specific pathway, and the value stored in that element in the matrix reflects some measure of connectivity. So for instance, the corner entries here correspond to the connection between left and right frontal lobes. We similarly have here a connection between the left frontal lobe and the left temporal lobe. And we also have a connection here between right frontal lobe and left occipital lobe. And these all have different numbers of streamlines, and this is reflected in their relative intensities within this matrix display. So whenever we're looking at these connectome matrix data, it's important to recognize that each of those elements corresponds to a specific pathway, and the value reflects some measure of connectivity for that particular pathway. And so the importance here is that if we can recognize that the process of deriving a measure of connectivity is equivalent for each of these elements within the matrix, it means what we can do is, for the purpose of this talk, is focus specifically on the techniques by which we derive such a measure of connectivity just for one specific pathway, because then to produce from that a connectome matrix is simply a matter of iterating over all possible pairs of grey matter regions. And obviously the actual analysis of those matrices will not be covered in this particular course. It happens later on in other subjects. So for a couple of years now, I've been trying to develop a framework by which I can express the kind of fundamental requirements that people need to be aware of when they're using diffusion MRI tractography. So there's a lot of different techniques out there. There's a lot of different complex methods with weird acronyms. I couldn't possibly cover the whole breadth of methods within a 30-minute session. So what I'm trying to do instead is present the things that you need to think about in order to critically assess whether or not a pipeline is robust, and what type of interpretation should be applied to the resulting data. And hopefully this will allow people to both make decisions about the pipelines that they wish to use, and also critique techniques that have been used through the literature. So the way I've managed to devise this is into this acronym track. So these are the 4 fundamental requirements that we're going to go through one by one. So the first one is fairly simple. Trajectories. Obviously, we are digitally reconstructing white matter trajectories through space. If we want to be able to perform any kind of reasonable interpretation of our data, we need these reconstructions to be accurate with respect to the underlying biology. So to achieve this, I've broken it up into 4 separate sections: estimating and following the local fiber orientation estimates, and then terminating and validating individual streamlines. So again, we'll step through each of these in turn. So in terms of estimating the fiber orientations at the local level, this is the responsibility of the diffusion model. So for every voxel in our image, we need estimates of 1 or more fiber orientations that the tractography algorithm can then sample from as it's generating the trajectory. But fundamentally, this isn't a component of tractography itself. This is the responsibility of the diffusion model. So there's, again, a lot of different diffusion models out there in the literature. Again, I can't cover the whole breadth here, but a few things to think about when assessing which of these models may or may not be appropriate for tractography, or what beneficial attributes they may have. So for different diffusion models, some estimate fiber orientations, some estimate diffusion orientations. For tractography, generally the former makes more sense, as it's the fiber orientations that we want to follow. With the prevalence of what's referred to as crossing fibers in the human brain, it's important to utilize a model that is compatible with that kind of fiber complexity. So that's shown in the rightmost column here, where we have two discrete fiber orientations in a single voxel, and the original diffusion tensor model is simply unable to represent this, but there's now a wide range of diffusion models that can. Ideally, we would like a measure of the dispersion of fiber orientation, but there's only a few models that are able to do this. Measures of uncertainty that we might want to encapsulate within our reconstruction, ability to interpolate orientations, because at every step of the streamline reconstruction, you're not guaranteed to be right at the center of a voxel. You'll be in between voxels, so you need to be able to interpolate between them. Again, a very quick and dirty summary. Different diffusion models will have different pros and cons in the context of use in tractography. I'm simply advocating that people should be aware and think critically about what these are. So secondly, following the local voxel-wise orientations to produce streamlines. So this is the responsibility of the tractography algorithm. Although the streamlines paradigm is conceptually very simple, there can be a lot of free parameters in there that can all influence the outcomes of the tractography reconstruction. So very quickly, there's been a few manuscripts that have looked at these kinds of things. So changing things like the step size, so the difference between subsequent vertices on the streamline, the angular threshold, so how tightly streamlines can curve. Both of these can have drastic influence on the outcomes on the resulting streamlines. You can have thresholds that influence where the streamlines can go and where they have to stop. Another one to be aware of is the order of integration. So you might hear first order, second order, fourth order. Basically what this means is that if you're generating a streamline within a curved trajectory, you want it to actually follow the curved trajectory and not underestimate that curvature, because that will result in deviation from the actual biological reality. Another one you'll see very commonly is the classification between deterministic and probabilistic streamlines. So what this distinction means is that if you see a large number of streamlines at a very specific location and then apply a deterministic streamlines algorithm, you will generate the same streamline trajectory every time. There's no stochasticity within the algorithm. Whereas for a probabilistic algorithm, each time you repeat that experiment, you're going to produce a slightly different streamline trajectory. And these are often discussed in terms of a sensitivity versus specificity trade-off. But it's also important to recognize this is not the only mechanism by which different tractography algorithms vary. And finally, on to the issue of terminating and validating streamlines. So I'll present these two together. Historically, while diffusion models tell us the fiber orientations and therefore the direction in which to propagate a streamline, they're not very good at telling us where to stop that propagation. And in part, this can actually be brought back to the diffusion signal itself. So what I'm showing here on the right is the fact that there's 4 different possible configurations of actual biological fibers within an image voxel. All 4 of these lead to an equivalent diffusion weighted signal, and yet in some of them the fibers terminate and in others they don't. So we can't very reliably tell where a streamline's tractography algorithm should terminate a streamline based on the diffusion signal alone. So an approach that's used fairly widely these days is to instead exploit anatomical MRI data that provides us good tissue contrast, and then we can then use segmentation of those tissues to control where streamlines stop, where they continue propagating, and also whether they constitute valid trajectories. So for instance, you can see in the image on the middle, these little yellow spheres correspond to the locations of streamline terminations, and you can see they're pretty much distributed throughout the whole brain parenchyma, which is not reflective of reality. Whereas when we use specifically the ACT framework on the right, you can see that the streamlines only terminate at the interface between grey matter and white matter. They don't terminate, for instance, in the middle of white matter where fibers are known to not synapse. They don't terminate in CSF, which simply wouldn't make physical sense. So that's trajectories. The next of the four is reconstruction density. So this is, again, an ubiquitous question in the diffusion MRI tractography community. How many streamlines should I generate? So the answer that I've derived myself for this is that fundamentally you don't want the precision of your experiment to be degraded by having not generated enough streamlines. So the basic trade-off here is that if you generate a very large number of streamlines, you're requiring more computation time. But if you then do like a repeatability experiment where you simply regenerate your streamlines, those two results are not going to vary very much. So you've at least got high precision. Whereas if you don't generate an adequate number of streamlines, sure, you're not using as much computational resources, but there's possibly a large amount of variability if you simply then regenerate your streamlines from scratch. So it's about finding the balance between these two. And fundamentally, the extent to which your results vary as a result of this imprecision depends on the specific measure that you're deriving from your data. So it really has to be specific to your own experiment. So the image I'm showing here relates to estimates of tract volume rather than connectome generation. And what it shows here is that the estimated volume of a particular white matter pathway in the corticospinal tracts in this instance can actually vary quite a lot depending on how many streamlines you generate. So obviously this is difficult to make robust biological conclusions. When it comes to connectome analysis, there actually hasn't been a whole lot of published work here. I kind of wonder if people are hesitant to have other people relying on an answer to this question that they've provided. So generally my advice is to actually do that kind of repeatability experiment yourself. So the third one, the A, is what I've called attribution. And this relates to the fact that in order to quantify connectivity for a specific white matter pathway, you have to be able to first define that pathway. Now, in the context of specifically connectome construction, this is usually thought of as, well, okay, you look at your two streamline endpoints and you say, which parcels should I assign this to? But as we've shown in our own work, this isn't necessarily a trivial operation. There's actually a number of different ways in which this could be done. And it's also been shown that the results of the network analysis that you get can actually change quite drastically depending on what mechanism you choose. So again, I won't go through the full list here. It's just something that I would urge people to be wary of when they're constructing a pipeline, when they're looking at someone else's pipeline, to think about how specifically is this being done? How is it that streamlines, for each streamline, you're making the decision of which part of the connectome it contributes to. So finally, the Q in the acronym is quantification. So this one I've split into two components. Firstly, primarily speak about what I've referred to as intra-subject quantification, and then I'll speak very briefly about inter-subject quantification. So we're constructing a connectome matrix. We need to derive for each edge in that matrix a measure of connectivity. Fundamentally, what should that measure actually be? What do we mean when we're trying to quantify this nebulous structural connectivity? So coming back to my original image, we have this issue where we've got a digital reconstruction of a white matter bundle. We have the underlying biological reality of that white matter bundle. And we want some scalar quantity of connectivity that tells us something about the role of that connection within the network. So the way that I've formulated this is, while it may not be explicit in a lot of network analyses, if you think about the data manipulations that are going on, fundamentally what we're looking at is the flow of information around the network. You know, we often analogize to computers in a digital network, that sort of thing. So when we're constructing these networks, it seems to me the most logical way to think about it is for each edge, you want a measure of the capacity of that specific connection to transmit information, to contribute to that information flow around the network. So what are the range of ways in which we can attempt to quantify this capacity for information flow? Well, probably the most common one is still the number of streamlines. It's been known fairly early on that this is this has limitations in terms of its quantitative capacity. There's a lot of particularly streamlined stratigraphy reconstruction biases that can have a heavy influence on this parameter. So there are people more so in the development community that frown upon it quite a lot, but you'll see a lot of applications that still use it. It's a perfectly legitimate metric to use. It's simply important that you explicitly recognize that that's the metric of connectivity that's being used. Another common one is to compute the mean of some quantitative value within the pathway, the most common of which would be the fractional anisotropy, or FA, from the diffusion model. So, for instance, one way by which you can do this is you can generate a voxel image of streamlines density for a particular pathway from that generator mask, and then sample the values of the FA metric within that mask. Obviously here, because you're relying on the FA measure, you inherit the limitations of that measure, you inherit the limitations of the diffusion tensor model. Similarly, if you use some voxel-wise metric other than FA, now there may be some quite valid applications of this sort of thing. If you're doing, for instance, machine learning classification, that sort of thing, you might want to simply sample as many different quantitative metrics as you can and make use of all of that data. But when we're specifically talking about network analyses that rely on this interpretation of information flow, there may be some voxel-wise metrics that are not good proxies for that interpretation. The one that I've myself worked on in my own research is what I've now come to refer to as fiber bundle capacity. Technically, what this actually quantifies physically is the intra-axonal cross-sectional area of the bundle. And the way that we achieve this measure is by enforcing consistency between the tractogram reconstruction and the information that we get from the raw diffusion-weighted imaging data. So there's a few methods now available that belong to this kind of class. We're still struggling on the right terminology, to be honest with you. But the basic premise is that if you look at the image in the middle, the streamlines density here in the bottom image in the middle, you can see that the streamlines density image is not a good match for the underlying fiber densities as shown in the left-hand image. And this is part of what contributes to streamline count being a poor quantitative measure. Whereas in the image on the right, what these methods do is they allow different streamlines to contribute more or less fiber density. And by running through an appropriate optimization, what you get is a tractogram that kind of recapitulates the distribution of fiber density throughout the white matter. And the interpretation that this enables is to look at the different weights that are ascribed to these streamlines within the tractogram. You isolate the streamlines within your white matter pathway of interest. So here I'm showing this using two grey ROIs on the left and right. And if we look at a cross-sectional cut through that bundle, we are summing the cross-sectional area of that bundle. So this measure scales with both the number of axons and their caliber, so their diameter. So we're not getting a genuine axon count measurement. We're not really able to do that yet with diffusion imaging, but this is hopefully a bit closer to a bit more faithful to the underlying biology than a raw streamline count. So the second component of quantification, inter-subject quantification, I'll only speak about this very briefly because some of it gets quite esoteric. The main point I want to make here is that there's a number of different features of the reconstruction that may vary between individuals. And you don't want these kind of latent confounds of your reconstruction to lead you to erroneous conclusions about differences between individuals. And it's important to recognize that this doesn't just apply to deriving a quantity of connectivity for a specific pathway and comparing that across subjects, but also these kind of higher order graph theoretical measures. So that's the acronym track. I hope this walkthrough was insightful in some way. I have my contact details here, including the forum for the software MRTrix3, where there's all sorts of discussions about the nuances of tractography if you're interested in having a look. And I've got all of the references from my talk there. Thanks, everyone. Enjoy the rest of the course.
So I'd like to begin by thanking the Education Committee and Chair for once again selecting our education course on network neuroscience for presentation at OHBM 2021. And also thank our wonderful lineup of speakers who will be participating in the educational course this year. My name's Andrew Zalesky. I'm from the University of Melbourne in Australia. And in my presentation, I'm going to focus on three key issues that are involved in connectome analysis, specifically connectome thresholding, the often overlooked topic of null models, and I'll wrap up with an introduction to comparing connectomes. So to begin, let's consider the topic of connectome thresholding. What is thresholding? In its simplest form, thresholding involves eliminating the weakest or potentially spurious connections from a connectome. So we start with a dense connectome, as is shown here on the left, the connection density of 30%. We can progressively remove the weakest connections until we arrive at a desired connection density. So you might ask, what are the actual benefits of connectome thresholding? And it's important to emphasize that thresholding is not an essential step. In fact, it's a controversial step, and analyses can proceed on connectomes that have not been thresholded. But that said, connectome thresholding can improve the visualization of connectomes. It can reduce the potential false positives within a brain network. It can simplify the interpretation of some graph measures. It can reduce the computational and storage burden, particularly for high resolution connectomes mapped at voxel and vertex resolution. And also in some recent work by Buchanan, it's been shown that thresholded connectomes can improve associations between connectivity and certain behaviors. On the flip side, there are some good reasons not to threshold a connectome. And one of those is that the threshold choice is typically arbitrary, and I'll go into the details of that in one of the subsequent slides. And also, as shown by Sivier, thresholding doesn't really impact some connectome measures to a large extent. And so the argument is, well, if it doesn't impact that much, why add this unnecessary complexity to the connectome mapping pipeline? As Sivier showed here on the right, if we compute path length, for example, in the unthresholded network, which is on the left, you then compute path length in the thresholded network, the actual values of path length are more or less the same. And so why add this added complexity to the processing pipeline? Once we've thresholded, if we decide to threshold our connectome, an additional step that can then be performed is binarization. And binarization is where we're only interested in the presence or the absence of a connection. We're not interested in the connection strength itself. And a disadvantage of binarization is that the connection weight, the strength of a connection, can provide rich information, and it can inform the topological structure of a connectome. And so by suppressing that information and only considering the presence or absence, we're essentially losing the ability to perform to gain insight into the network. The advantage of binarization is that this variation in connectivity strength can be due to noise, or it could be due to methodological confounds. And by binarizing, so removing that information, we alleviate the effect of noise and these potential methodological confounds. So to summarize, with a binary connectome, we only have, as I've shown there on the right, ones and zeros in the connectivity matrix, one representing the presence of a connection, zero the absence of a connection. So here's a conceptual example to illustrate the process of thresholding in a toy network. In this toy network, we have 3 connection weights. They're either strong, moderate, or weak. And we start with an initial connectome with a connection density of 26%. The connection density is just the total number of connections in the network divided by the total possible number of connections in the network. This is the possible number of node pairs. And so at the first step here, we can remove the weakest connections, and this yields a resulting network with a connection density of 20%. So you'll see here all of the weak connections have been removed. Then we can go one step further and not only remove the weak connections, but also the moderate connections. And so the thresholded network only comprises strong connections. And this final network has a connection density of 13%. Notice here that one of the nodes becomes fragmented. It's disconnected from the network. And this is a problem for some graph analyses. And it's also biologically implausible that we have nodes that are disconnected from the broader network. And I'll show you some approaches to overcome this issue in a moment. There are 2 broad classes of thresholding methods: density thresholding and weight thresholding. With density thresholding, we prescribe a desired connection density, in this case 20%, and we successively remove the weakest connections until we arrive at our desired connection density. Now here I've noted the brain connectivity toolbox function, which performs proportional thresholding or density thresholding, as it's also known. Throughout my slides, I'll present particular functions so that you can use these in your own work. These are functions from the brain connectivity toolbox. We also have weight-based thresholding. With weight-based thresholding, rather than prescribing a desired connection density, we instead prescribe a weight, a cutoff or a threshold on the weight. So in this case, we say we only keep connections that comprise 3 or more tractography streamlines. And we see here in the thresholded network, the connection here between these 2 nodes that comprises only 2 streamlines is removed from the network. The crucial point here is that there's a one-to-one correspondence between these 2 thresholding methods. So if you give me a certain connection density, say 20%, I can always tell you the corresponding weight threshold that will achieve that connection density, and vice versa. For any given desired weight threshold, a corresponding connection density can be found. And so you can ask, well, why make a difference between, why do these 2 methods exist if there's a one-to-one correspondence between them? The difference is that these methods differ when applied to group studies or when comparing individuals. And to illustrate that, here I've considered connectomes for 2 individuals, for Jack and Alice. And as you'll notice here, Jack's connectome comprises a greater number of strong connections. These are denoted by the thicker lines relative to Alice. And so if we apply weight-based thresholding, in this case, we remove the moderate and weak connections, and the thresholded network here is shown on the right. And because Jack's connectome comprises a greater number of strong connections, the thresholded network differs in connection density, or in other words, the number of connections between Jack and Alice. And the problem with this is if we were then to go on and do subsequent compute certain topological measures, if we see differences in those topological measures between Jack and Alice, these might trivially be due to the fact that the number of connections between Jack's and Alice's thresholded network comprise a different number of connections. So this is a potential confound. So you might say, okay, well, if weight-based thresholding is associated with this confound, perhaps we should move to density-based thresholding. And indeed, if we use density-based thresholding, remember with density-based thresholding, we prescribe a desired connection density, and this by definition will ensure that the thresholded networks of Jack and Ellis comprise exactly the same number of connections, and therefore we alleviate this potential confound, the confound being the difference in connection density between the individuals. The concern with density-based thresholding is that we see for the case of Ellis, to ensure that Ellis has the same number of connections as Jack, we've had to add in these moderate connections between these nodes. So these moderate connections, because they're weaker in strength, they're potentially spurious. So it could be argued that where the process of matching connection density between the two individuals has required spurious connections to be added to to Ellis's network, which is also a concern. So which of these two approaches should we use? To illustrate this in a little more detail, here I'll consider the example of schizophrenia. For the case of functional connectivity networks, it's well established that functional connectivity is generally reduced in individuals with schizophrenia compared to healthy controls. So in the case of weight-based thresholding, if we discard functional connections with a correlation coefficient less than 0.2, the thresholded network in the patients will have a connection density that's substantially lower than the healthy control group. And this introduces that confound that I've discussed. If we then go on and do further graph analysis, the fact that controls have a higher connection density can confound analyses of certain topological properties. So we say, okay, let's go to density thresholding instead. And so if we prescribe a density threshold of 80%, we continue to remove connections until we achieve that density threshold, we see that the minimum connection weight in the patient is substantially lower, 0.31, relative to the healthy control, which is 0.42. So we've had to lower the bar for the for the patient to ensure that we have a matching in connection density. And once again, by lowering the bar, we could potentially be adding in spurious connections. So if you're interested in this, in learning further details about this, check out the work by Van den Heuvel in 2017, which proposes some solutions to this issue. Another key concern for thresholding is the choice of threshold, and this is often chosen arbitrarily. We don't know what the ground truth connection density of a connectome is. Is it 10, 20, 30%? So one approach to overcome this is to consider the sensitivity of the particular network analysis across a range of thresholds. And in fact, if we're looking at a particular network measure, such as path length, as I've shown here, we can evaluate path length over a range of thresholds and then consider the area under the curve of the function of that particular network measure as a function of connection density. And this takes away the dependence on one particular arbitrary choice of threshold. This also relates to the topic of topological data analysis. Check out the work of Sizemore in 2019. And an alternative approach to avoid arbitrary selection choice is referred to as multi-threshold permutation correction. I won't go into the details of that here, but this involves essentially performing a statistical test at each threshold and testing for rejection of the null at threshold, and thereby avoiding an arbitrary threshold choice. Another important thresholding approach, in addition to density and weight-based thresholding, is referred to as consensus thresholding. And the idea here is that we aim to threshold based on finding a consensus of edges across individuals. And in this particular example, we have three individuals, Jack, Ellis, and Bob, and we only keep connections that are moderate or strong in two of the three individuals. So we see here that in the case of Jack, the connection between these two nodes is strong, but it doesn't appear in Jack's thresholded network. And the reason for that is that the connection is weak or moderate in Ellis and Bob. So for a connection to appear in the thresholded network, it needs to be moderate or strong in two of the three individuals. The disadvantage of this approach is that we need to choose two particular thresholds. One is the weight threshold, which is in this case moderate or strong, as well as the group threshold, in this case is 2 of the 3. The other thing to consider here is that consensus across a group of individuals doesn't necessarily mean that the connection is not spurious. I mean, we can have systematic errors that appear across a group of individuals. I'll now briefly touch on some alternative or some newer thresholding approaches. Thresholding can also be cast as a problem in statistical inference. If we're looking at functional connectivity values that are mapped using correlation, we can convert the correlation coefficient to a z-score, and then compute a p-value. The thing to be careful here is that because of the autocorrelation in bold data, these z-scores are typically inflated, and therefore p-values are too optimistic. And there are some certain corrections that have been proposed to adjust for this, to adjust for the autocorrelation in the bold. Check out the references that I've shown at the bottom there for further details. What I've discussed so far are only local thresholding methods, global thresholding methods. There are also local thresholding methods that can emphasize certain topological properties in the connectome. These include the disparity filter and multi-resolution thresholding. These are not as common in connectomics. If you're interested, check out the references that I've listed there. And finally, an important approach to thresholding is referred to as the minimum spanning tree. This ensures that the resulting thresholded network isn't fragmented. So the basic strategy is that we define a backbone, a connected backbone, and then add further connections to this backbone, which was referred to as the MST, until we achieve a desired connection density, and thereby avoid this problem of having fragmented nodes that are not connected to any other part of the network. So to summarize, connectome thresholding involves eliminating the weak and potentially spurious connections from a connectivity matrix. It's important to emphasize that thresholding is not essential, but it can alleviate false positives, ease computational burden, can simplify the interpretation of some graph measures, and certainly improves connectome visualization. Here I've discussed the two most common approaches to thresholding, those being weight and density thresholding. I've described the equivalence between the two and how these differ in group studies. I've also emphasized the importance of evaluating a range of thresholds to ensure that any analysis isn't dependent on one arbitrary threshold choice. So there's some further readings listed here, and also remember the two brain connectivity toolbox functions that are available to perform weight and density thresholding. So in the next part of the presentation, I want to move on to null models, which are often overlooked in connectomics, but these are crucial to establish whether certain topological properties appear in a connectome, more so than that would be attributed to chance or the effects of noise and confounds. And I want to motivate the importance of null models using a particular example of path length. And this will be discussed in one of the next presentations in the course. But for here, we want to consider a simple network, as I've shown here, a simple connectome. And here the path length between these two yellow nodes is 2 because there are 2 hops. There are 2 connections that need to be traversed between these 2 nodes. So a question that can be asked here is the path length of 2 for this particular network, is it normal, is it typical, or is it particularly short, or is it particularly long? And to address that question, we need a benchmark network. We need some kind of a benchmark, or what we refer to as a null network. And a simple null network here is a random network that comprises the same number of nodes and the same number of edges as our observed network on the left, our observed connectome. And in a random network, the average path length can be computed with this analytical formula. We plug in the number of nodes, in this case 9, and the number of edges, in this case 15, and we see that the average path length in a network of this size is 2. And so the conclusion that we draw here is that the path length of 2 in our connectome is typical. So our network is not particularly conducive to short path lengths, because we would expect that kind of path length in a random network with the same number of nodes and edges. Now let's turn to a lattice network. And in this lattice network, we have 20 nodes and 40 connections. And once again, we can compute the average path length, and we find that the average path length in this lattice is 2.5. So on average, we need to take 2.5, we need to traverse 2.5 connections to get between any pair of nodes. And we can ask the same question: is 2.5 long? Is it short? Or is it typical for a lattice network? So once again, to address this question, we turn to our benchmark network, in this case a random network. We once again plug in the number of edges and the number of nodes, and we see that the average path length for a, the average path length for a random network is of this size is 2. And so what we can say is that a lattice network, because the average path length in a lattice network is 2.5, lattices are not particularly conducive to efficient routing. So paths are longer in lattice networks compared to random networks. Now, by random network here, I mean a network where we add connections to pairs of nodes at random until we achieve the desired number of connections. So in this particular case, the null network or the benchmark network was matched to our actual connectome, to our actual network, in the number of nodes and the number of connections. But you might ask, why do we stop there? Why don't we introduce other properties into our benchmark network, into our null network, such as node degree and connection length? And to a degree, it is an arbitrary choice as to what properties we preserve in our null network. So looking at this a bit broader, we can define a set of all possible we can draw null networks from the space of all possible random networks. And this is a pretty easy null to reject, and it's not particularly useful. If we constrain the null space a bit more, we can say, well, we want to ensure that our benchmark or our null network preserves the number of nodes. If we go to the unit concentric circle, we say we want to preserve the number of nodes and edges. And so as you preserve more and more properties in our null network, the number of null networks begins to shrink until we eventually converge on our true connectome. Now, in connectomics, it's typical that we preserve 3 properties of our observed connectome, those being the number of nodes, the number of edges, and the node degree. So that's the green concentric circle that I've shown here. In some recent work, there has also been interest in not only preserving these 3 properties, but also connectome geometry, namely the length of fibers. And this gives us an even more constrained null. That's the salmon-colored null here, the null space shown here, and allows us to test hypotheses regarding geometry. So what do we do how do these actual null networks work in practice? So in practice, what we would do, we sample thousands of null networks from a certain null distribution using multi-Keilholz simulation, and then we can compute a normalized measure. So once again, if we're looking at path length, the numerator here is the path length in our actual network. We sample networks from our null space. So from, in this case, it's being the salmon-colored null space, and we average the path length across each of these samples, and we take the ratio. So if the ratio is greater than 1, we see that path lengths are greater in our actual network compared to the null. If the ratio is 1, path lengths are equivalent, and if the ratio is less than 1, we see that path lengths are particularly short in our actual network compared to the benchmarks. We can also perform statistical inference and hypothesis testing. I won't go into the details of that here. In practice, the way this is done, once again, as I've said, we usually preserve only node degree, number of edges, and number of nodes. And this can be done using an approach called Maslov-Snepen rewiring. Step 1, we take our observed connectome, find a motif, such as I've shown here, where U is connected to V, I is to J. We rewire that, as I've shown here on the left. So U connects to I, V to J. And we repeat this step hundreds and hundreds of times until our network is randomized. And if you think about this for a while, you'll see that this will guarantee to preserve node degree and also the edges, number of edges and number of nodes. And then we can compute our measure for the randomized network, and this gives us 1 sample from the null distribution. This approach can't be applied to weighted and side networks, but there are some heuristics that are available, and check out the work by Rubinov and Spawns for further details. So here's some examples of Maslow-Stepan rewiring in practice. This is for the macaque connectome and the mouse connectome. The left here is the actual macaque connectome. The right here is a sample from a null network that was generated using Maslow-Stepan rewiring. And here on the rightmost, we are showing the ratio of clustering in the real network, in the real macaque connectome, to the null network. And we see that clustering, the normalized clustering, or the ratio, is greater than 1, suggesting that clustering is greater in the macaque connectome compared to the Maslow-Stepan rewired networks. Path length, the ratio is approximately 1, so the path length is equivalent between the actual and the null. And small worldness is greater than 1, suggesting that these are small world networks. We see similar behavior in the mouse connectome as well. There is also the concept of geometry preserving null networks. I won't go into the details of this. Check out the work by Roberts, as well as Klim and Betzel. The idea here is that we want to ensure that our null network preserves fiber lengths. And what we see is that if we preserve fiber lengths in our null network, there are particular topological properties that become closer in the geometry preserving null to our connectome compared to the non-geometry preserving null. So if you focus on the bottom right of this slide, where I've shown clustering coefficient as a function of connection density, the blue is the clustering coefficient in our connectome. The red is the traditional Maslow-Stepan rewiring, so that we see that there's a large gap between the connectome and the non-geometry preserving. But once we add geometry back to our null, the null networks become closer to the actual connectome. And this suggests that the geometry of the connectome, namely fiber length, to a certain extent can explain the network's clustering coefficient. There are also the concept of generative models, which are related to null models, and this is where we use basic mechanisms to generate networks in silico that resemble connectomes. And so Vertes defined 3 particular models that are based on simple mechanisms to generate networks in silico, and found that model 3, the economical clustering model, provided the best match for functional networks. In this case, we define a probability of finding an edge between 2 nodes, and this is based on the distance between the 2 nodes. So we want to encourage connections between nodes that are spatially in spatial proximity to each other, as well as a topological rule. And work has shown that the matching index, which indexes homophily, is the best performing rule. And this is where we want to add connections between nodes that share similar neighbors, that are connected to similar neighbors. Check out the work by Betzel for further details about generative models. So to wrap up the topic of null models, I want to emphasize that null models set expectations for network organization under certain benchmarks. Typically, we generate null networks that preserve the nodes, edges, and the node degrees, and this is done using Maslow-Stepan rewiring. More recently, there's been an emphasis on focusing on geometry preserving nulls, and we see that geometry can, to a certain extent, explain some of the topological properties that we see in connectomes. I've also briefly touched on generative models, where we can grow networks in silico based on very simple geometric and topological rules. Once again, there's some further reading there, and I've listed the BCT functions for the Maslow-Stepan rewiring algorithm, as well as the function for generative modeling. So finally, I want to touch on comparing, on the topic of comparing connectomes. And this is the case where we've mapped a group, mapped connectomes for a group of healthy individuals, as well as a group of patients or a treatment group. And we want to either identify connections that differ between the 2 groups, or that are modulated by the treatment. This is referred to as connectome-based inference. Or alternatively, we may want to consider the connectomes as a feature space and predict clinical outcomes or treatment responses using this connectome-based feature space. The challenge here is that the dimensionality of connectomes is huge. We typically have hundreds of nodes and thousands of connections. So I want to start by focusing on connectome inference, and inference can be performed at 3 distinct levels. The simplest form of inference is global comparison. This is where we only have one particular measure for each individual, for each connectome. So these can be measures such as global efficiency, the modularity coefficient, Rich club, and so on. The difficulty of this is we can't localize an effect to a particular node or a particular connection. But it is quite easy to deal with these global comparisons. We can use classical statistical tools. If we move one step up, we can consider node level comparison. This is where we're comparing measures such as node strength, node efficiency, and so on. This allows us to localize differences between groups or to localize an effect to a particular node, to a particular region of the network. And what I'm going to focus on here is the level one above this, and this is where we're performing connection level comparison or connectome-wide studies, and this is where we're looking at the comparing connectivity strengths. This could be, for example, functional connectivity as measured using correlation or the streamline count. This is also referred to as a connectome-wide association study, and in its most basic form, this involves testing the same hypothesis at each of the possibly thousands of connections comprising a connectome. So we could use any of a number of models, t-test, f-test, ANOVA, and so on, fit that model to each connection, and this ensures that each connection has a test statistic and a corresponding uncorrected p-value. And then we need to correct for multiple comparisons across the set of all connections comprising the network. There are various generic methods for correcting for multiple comparisons, you probably already know. Methods such as Bonferroni correction, which are probably too stringent unless the sample size is huge. There's also the false discovery rate, which is a more appropriate method for connectome inference. The difficulty with these generic approaches is that they don't take into account the inherent properties of a connectome. And what I mean by that is that what we generally see is that effects of interest are generally clustered within each other. So between group differences are found within subnetworks that are focused on a given set of regions. And we see this across a range of disorders as was shown there on the left. So it's more likely that we have scenario 2, where we see subnetworks, where we see the signal of the effect of interest within a subnetwork as opposed to being dispersed across multiple disconnected connections in the network, as I've shown here in scenario 1. Now, the key point here is for methods such as FDR and Bonferroni, they don't offer any advantages for scenario 2 over scenario 1. In fact, they'll offer the same power, statistical power in both scenarios. But there has been some methods that have been developed that will improve statistical power by leveraging this particular property that effects form connected subnetworks. So one such approach is the network-based statistic. I won't go into too much details here, but this involves setting once we have a test statistic network, so a test statistic at every edge, we apply a threshold based on the minimum meaningful effect size, and then in the resulting thresholded network, we search for subnetworks, or in other words, connected components. So in this example, we see a connected component of size 5, and then we can apply permutation testing to compute a family-wise error-connected p-value for this subnetwork. Briefly, this involves permuting labels between, if it's a patient control study, permuting labels between patients and controls, computing the test statistic network for this permuted data, thresholding, and then looking for the size of the largest subnetwork in the permuted data. And here I've shown a few permutations, and I've also shown the empirical null distribution of the permuted data. And we see that the actual network, so in other words, a subnetwork of size 5, is exceedingly rare in the permuted data, and so we can compute a p-value based on the proportion of permutations where the size of the largest component was greater than 5. In this case, it's 0.02. So we can reject the null and claim that this subnetwork is significant for this between group difference. There are other methods for connectome-wide analysis studies. I'm not going to go into the details here, but check out the references. These include matrix factorization, CCA, multivariate distance matrix regression, and sum of power score. So in the last part of the talk, I now want to move to connectome-based prediction as opposed to inference. And this is where we aim to build predictive models to predict individual characteristics based on features of the connectome. And this is, we might want to predict clinical outcome, treatment response, diagnostic status, and so on. And this is usually done within a cross-validation framework. So as I've shown here, we would take the feature space to be the upper triangle of each individual's connectivity matrix. We define test and training sets, and then we train a model to predict our desired outcome and evaluate accuracy using a test set. So this is some work that was done on by Dady, which evaluated various generic prediction models, and was found that L2 regularization, such as and ridge regression, yielded the highest prediction accuracy. So the more to the right, the higher the prediction accuracy in this plot. There are alternative or more connectome-specific predictive modeling, such as CPM or connectome-based predictive modeling. Check out the work of Shen for further details. Some particular tips for connectome-based prediction. The first one is that there is some utility in transforming connectivity matrices to a so-called tangent space. This can improve prediction accuracy to a small extent, as I've shown here on the plot, where we're comparing tangent relative to correlation and partial correlation functional connectivity measures. Check out the work of Varequai and Dady for further details. The issue of sample size is crucial. We know that the error bars on accurate estimates are large for small sample sizes, so it's imperative to consider large sample sizes for prediction-based analyses. Some other general tips is to consider k-fold as opposed to leave one out, cross-validation. Use the error end of the curve to evaluate accuracy. Be cautious of data leakage, so where the training data can sometimes appear in the test data, which can inflate prediction accuracies. And evaluate, if possible, evaluate accuracy across multiple sites and potential scanners to demonstrate the generalizability of the prediction model. To wrap up, here's some software to perform connectome analyses. In this presentation, I've largely focused on the brain connectivity toolbox, which there are both MATLAB and Python versions of the toolbox. As you can see, there's a wide range of software that is available to you to perform network inference as well as thresholding and to generate null models. That's all from me. I just wanted to once again mention that we are running 2 interactive sessions after the course. We look forward to seeing everyone at the 2 interactive sessions. I've shown the time here. This will provide you the opportunities to interact with our speakers, to ask questions, and also to provide feedback on the course. I wanted to also draw your attention to, if you wanted further information on network analysis, check out our book. This is a resource for brain network analysis. This is written by Alex, myself, and Ed Bullmore. So I'm going to stop there. Thanks again for your attention, and I look forward to meeting everyone at one of the interactive sessions. Thank you.